Description of embryo development in cassava. Cassava is in a unique position among the most important crops for humanity. 20 years ago, the first molecular map was published with research conducted here in Colombia. Cassava genome was sequenced and published several years ago. In contrast to these advances, there is some basic and critical information that is still needed even to implement many of the new technologies, such as marker-assisted selection and genomic selection. The limited information available on the reproductive biology of cassava is an example of the gaps in basic knowledge of this crop. In the double haploid project, great efforts had to be invested to fill this lack of information on reproductive biology of cassava. Another module describes, for example, the pollen 2 growth rate that turned out to be slower than anticipated. This module presents all the information relevant to embryonic development. Knowing the chronology of each stage of embryonic growth is fundamental, for example, in the development of protocols for the production of double haploids by genogenesis, as well as in broad crosses. In both strategies, as there are problems in the development of the endosperm, the embryo eventually aborts, so early embryo rescue should be employed. At the beginning of the project, however, we did not have a clear idea of what embryo development was like in normal cassava crosses.
Por varios años realizamos secciones histológicas en frutos cosechados en distintos tiempos, luego de la polinización, para poder entender mejor la cronología del desarrollo embrionario de la yuca.
In this module, we will initially describe the conventional use of the microtome in the preparation of samples for analysis. In another module, a new technology based on the use of the vibrotome for sample preparation is described. We have integrated the conventional histological methods and the use of the vibrotome in order to understand the embryonic development of cassava, which will be presented in the second part of this module. Cassava researchers have a limited knowledge of the reproductive biology of the crop. Breeders have been making controlled pollination for decades, but there is very limited information on how the embryo develops and the timing of the key stages of embryo development. The following images provide a chronological description of seed development in cassava. Illustrations of the embryo sac in cassava are initially provided. Then, the different stages of embryo development that could be documented after hundreds of sections analyzed are presented. The final portion of this video presents the appearance of seeds and embryos as they mature. Early stages of embryo development is the most relevant information required for developing a doubled haploids protocol. It is also the kind of information that was not available in the literature. Description of the embryo sac in cassava. For several years, the double haploids team has evaluated hundreds of histological sections to document the embryo sac in cassava pistil, identify the key stages of embryo development, and what was very relevant for the team, the timing at which these stages were attained. These are transversal sections of cassava embryo sacs around anthesis day. The intense pigmentation in the center of this photograph is the pollen tube growing through the synergid cells. In this transversal section of the embryo sac, the egg cell is clearly identified. The accumulation of starch granules around this cell is also conspicuous in this transversal section of the embryo sac. This is a longitudinal view of an embryo sac. It shows the large egg cell. Starch granules around the egg cell are also visible. This is another longitudinal view of an embryo sac in cassava. At its center, the image of a central cell of the nucleated endosperm has been captured. Early stages of embryo development in cassava. Embryo development in cassava is slow and, as it turned out, surprisingly irregular. 7 days after pollination, a pro-embryo with just 4 cells could be identified at Este University. This is the earliest image of an embryo at pre-globular stage that the doubled haploid team could document. The section was prepared from a fruit collected in cultivar HMC1, 12 days after pollination. At Este University, a pre-globular embryo could be observed in samples collected 14 days after pollination. Nucleated endosperm is actively growing. Another illustration of an embryo at the early globular stage obtained with the use of the vibratome, but this time observed with Nomarski's differential interference contrast optics. The sample was 130 micrometers thick. Embryo development is still at the initial stages, yet the fruit was collected 21 days after pollination. Comparing this with the previous photographs illustrates how irregular embryo development in cassava can be. This photo illustrates a well-developed globular embryo, 19 days after pollination. It offers a sharp contrast of the previous image, which showed an embryo at its early stages of development, but from a fruit harvested 21 days after pollination. A globular-shaped embryo can be clearly spotted in this image. The photograph shows the intensely pigmented outer integument that clearly contrasts with the inner integument. The sample was prepared with the vibratome, but in this case is seen through an ordinary light microscope. The section is 60 micrometers thick and shows a well-developed globular embryo, which was taken 24 days after pollination. The beautiful image of this 24 days old globular embryo demonstrates the advantage of preparing samples with the vibratome. The section is 130 micrometers thick and was observed through Nomarski's differential interference contrast optics. The position and size of the suspensor is clearly visible. In the image, a cleavage in the shape of the globular embryo is just beginning to occur. Eventually, the cleavage will accentuate and give rise to a heart-shaped embryo. This section shows a well-developed heart-shaped embryo. The fruit was collected 22 days after pollination. The fruit that originated this section was collected 24 days after pollination. The embryo is emerging from a torpedo shape into the cotyledonor stage with the 2 cotyledons clearly distinguishable. This is an embryo 29 days after pollination. Cotyledons are very large. The shoot apical meristem is beginning to appear. In the opposite end of the embryo axis, the root apical meristem is already developing. Chronology of embryo development in cassava. Pollen tube growth has been found to be slow and variable. Detailed studies, described in another video within this series, indicate that the earliest arrival of tubes at the embryo sac occurs about 24 hours after pollination. However, in most cases, it takes considerable time, up to 96 hours, for the pollen tubes to get in contact with the sac and eventually fertilize it. 7 days after anthesis, personnel at Este University could detect the first evidences of cell division within the fertilized egg cell, which then gives rise to the zygotic embryo. Embryo development is slow and its rate highly variable. Preglobular or early globular stages of the embryo have been found from 12 throughout 22 days after anthesis. This is a very surprising and puzzling finding. Since cassava does not depend so much on sexual reproduction as grain crops, there has been no evolutionary pressure to fine-tune and maximize the efficiency of sexual reproduction in this crop. Later stages of embryo development, however, are shorter and more uniform. Globular embryos have been found between 19 and 26 days after anthesis. Heart-shaped embryos have been found starting at 22 days after anthesis. Since the scope of this research focused on earlier stages of embryo development, few samples beyond 24 days of age are available, and therefore it cannot be said with precision the latest age at which heart-shaped embryos can be found. Torpedo-shaped and the transition into cotyledonal embryos was found to occur between 24 and 29 days after anthesis. Late stages of seed and embryo development in cassava, starting at 20 days after pollination. Cassava breeders have had a good understanding of the different stages of flower development, particularly around anthesis day. Bracts retract the day after anthesis. When fertilization does not occur, the developing fruits may remain attached to the plant for few days. Eventually, however, a drastic abortion rate has been observed between the first and second weeks after anthesis. After the second week, there is very little fruit abortion. Fruits mature, dry and open 2 to 3 months after anthesis. Environment and genotype influence the time required for fruits to mature, dry and release the seeds. These are 20 to 25 days old seeds. More often than not, embryos cannot be detected with the naked eye at this age. These images illustrate different embryos from dissected seeds. The fruits from which these embryos were extracted were collected 4 to 6 weeks after pollination. These are 32 to 38 days old embryos. On the left is the remains of the developing seed from which one of these embryos was extracted. Embryo size differences gradually reduce in later stages of development. These are 30 to 32 days old embryos. Notice the large variation in the size of embryos of about the same age. Embryos develop slowly during the first 30 days. The rate of development, however, is surprisingly uneven. This is a 6-week-old embryo at its natural position inside the cellular endosperm.